Good morning, everyone. My name is Raksheet Reddy Gummakonda, and today I'll be presenting my research titled How Does Vitamin C Change Blood Antioxidant Capacity? This project uses a simple, low cost chemical method, idiomatic titration, to measure how diet can change human blood chemistry within just 1 hour. We often hear the phrase, how what you eat affects your body, but very few people know how fast that actually happens. So I wanted to ask a simple but powerful question. Can one dose of vitamin C produce a measurable biochemical change in the blood within just 1 hour? To test this, I used idiomatic titration, a classical, inexpensive chemistry technique, on my own blood samples. This allowed me to track rapid shifts in antioxidant capacity without any high-end equipment. What I found is that even a basic dietary choice can cause immediate measurable changes in blood antioxidant power, and that meaningful human health data doesn't always require expensive technology. To understand why this matters, let's look at a global problem: oxidative stress. Our body continuously produces oxidative reactive species, or ROS. When ROS accumulates, they damage DNA, proteins, and cell membranes. This process drives major chronic illnesses like heart disease, diabetes, cancer, and even aging. The WHO, the World Health Organization, reports that 74% of global deaths come from chronic diseases in which oxidative stress plays a major role. So the question becomes, if diet can influence antioxidant levels, how quickly can it influence them? And more importantly, can a student research project measure these changes safely and cheaply? Here's the gap. Scientists want to know that vitamin C is one of the scientists know that vitamin C is a main antioxidant found in blood and that its levels rise after ingestion. But what we don't know is that, especially at the student research level, is how quickly antioxidant capacity changes in real time, whether low-cost chemical methods can capture this accurately, and whether self-sampling can produce reliable physiological data. So my project specifically targets this gap. Can we measure immediate biochemical changes using accessible tools? So I approach this using a classical technique, idiomatic titration, where vitamin C neutralizes iodine so that more vitamin C present in plasma, the more iodine it consumes before the blue starch iodine complex forms. This gives us a direct measurable numeric of antioxidant capacity. In other words, more vitamin C, more iodine needed, stronger antioxidant powder. It's simple, inexpensive, and surprisingly effective. Here's the chemistry in one slide. Vitamin C, or absorbic acid, donates electrons. Those electrons reduce iodine from iodine 2 to iodine minus 1. As long as vitamin C is present, iodine cannot form the blue starch iodine complex. So the endpoint, the moment the mixture turns blue, only happens after all the vitamin C has reacted. This is what makes idiomatic titration such a powerful proxy for antioxidant capacity. The key takeaway here is that the higher concentration of vitamin C means a delayed endpoint and a larger volume of iodine required. This chemical mechanism is well understood and makes the method robust and quantifiable. Here's the experimental design I used. I collected a baseline sample after fasting, then drank 200 ml of fresh orange juice, and exactly after 1 hour, I collected a second blood sample. I then separated the plasma using a centrifuge, and then for each sample, I added acid and a starch indicator. Then I titrated with 0.1 mole iodine until the blue endpoint persisted. Each titration was done in triplicates for accuracy. And then finally, I calculated the vitamin C concentration in mg per 100 ml of plasma. These are the photos of my actual procedure. This was a fully hands-on experiment, including my self-sampling under safe, supervised conditions. Before showing you the numericals, here's what I visually observed during the titration process. For the baseline plasma sample, the endpoints appeared almost immediately within just 2 to 3 drops of iodine. This basically indicates very low antioxidant activity. But after the vitamin C intake, the plasma required 10 to 14 drops before turning blue. There was a clear delay, meaning vitamin C was actively reducing iodine. This volume rose about 0.005 ml to 0.015 ml, so basically a threefold increase. Even qualitatively, the difference was obvious. The post vitamin C plasma persisted the blue color much longer, confirming increased reducing capacity. And top it off, triplicate measurements were consistent, showing reliability. Now let's look at the actual numbers. First, iodine consumption. The baseline plasma needed only 0.005 ml of iodine to reach the endpoint, whereas after vitamin C, it required 0.015 ml, which is a clear threefold increase in, antioxidant activity. Second, plasma vitamin C concentration. Using the titration calculations, baseline vitamin C was 0.88 mg per 100 ml. Post ingestion, it rose to 2.64 mg per 100 ml. This represents a 200 percentage increase moving from the normal low range to the upper physiological limit. These two independent metrics, iodine volume and calculated concentration, both support the same conclusion. Vitamin C significantly boosts plasma antioxidant capacity within just 1 hour. Like any project, this study has limitations. It involves a single participant, myself, so it reflects individual physiology, not population trends. Idiometric titration is not specific to vitamin C. It measures total reducing agents, and orange juice contains other antioxidants, so there is a mild confounding. I only tested one time point, so I couldn't capture a full absorption curve. And manual titration also introduces small subjective errors in endpoint detection. These limitations help define what future work could address. Moving on, here's what the results tell us. First, plasma vitamin C and antioxidant powder both increased significantly, about a threefold within just 1 hour. This shows that diet can rapidly and measurably alter blood chemistry. Second, it validates iodometric titration as a reliable, low-cost method for human biochemical analysis. Looking ahead, there are several exciting extension pathways, expanding the sample size to explore biological variability, adding more time points to build a full vitamin C pharmacokinetic curve, comparing natural versus synthetic vitamin C sources, and then validating findings using advanced assays like FRAP, ORAC, or HPLC. What began as a small experiment opens the doors to larger studies in nutrition, antioxidant biology, and accessible human health monitoring. To conclude, I want to leave you with three key messages. First, nutrition is a global health issue. Antioxidant stress drives chronic diseases worldwide, and this project shows how even simple dietary choices can strengthen biochemical defenses quickly. Second, scientific innovation doesn't always require expensive technology. A simple titration technique generated meaningful human health data. Third, small scale experiments contribute to big global issues like chronic disease prevention, public health nutrition, health equality, and science democratization. Ultimately, this project demonstrates that accessible science can still deliver powerful insights about human health. Thank you so much for listening. I'll be happy to take your questions.